Hi, my name is Paul Scholz and I am the technical lead of the genome editing development at BRAIN. And today I would like to give you a short overview and some insights into our BRAIN Engineered Cas technology. When we are talking about genome editing, we have a technology that has revolutionized biological and pharmaceutical research over the last years. Because if you know what you're doing, you are able to edit the genome of almost every living organism in a way that's way more fast, efficient and cheap compared to other technologies that have been there already. So as BRAIN, as a white biotech company, of course we also have huge interest in genome editing, and during the last years we have done a lot of experiments and gathered a lot of expertise using these tools, for example in different bacteria, different fungal systems, yeast systems and also mammalian cell lines like cat cells and human cells, for examples. What we are able to do with this technology is that we are able to upregulate and downregulate genes, we are able to knock out and knock in genes, we can do multiplexed edits and use genome-wide libraries. So overall, we have gathered extensive experiments using genome editing tools. But the big downside of these tools is that we are not allowed to use this on a commercial level because of the still ongoing patent war and the complicated IP situation Michael already discussed in his presentation. Beside from these business-related issues, there are also technical limitations to the classical CRISPR genome editing tools that are available already. For example, if we look at Cas9, which is the most prominent CRISPR-Cas nuclease, we have things like on and off target effects, which means that we will generate an unwanted alteration in the genome of the cell that we would like to edit. And if we're thinking, for example, about gene therapy in human cells or in humans, of course, unwanted changes are not acceptable at all. Then we have Cas9 as a protein itself. So it is a very big protein, has a very big size. And this can make it quite hard to deliver the Cas9 protein efficiently into your target cell. Furthermore, we have efficiency or variable efficiencies across different organisms, across different cells, and also across different loci inside the same cell, which makes it almost impossible to predict the outcome of your experiment prior to your actual experiment. Furthermore, we have things like possible immune reactions because the Cas9 protein was found in a human pathogenic organism. Beside from a lot of these technical limitations, there's also the business side, which is the complicated IP situation with the ongoing patent war and the legal and financial uncertainties for all the users. So if we could have a wish list for an alternative Cas protein, a different one that would bypass all of these limitations, how would this look like? So of course, we would have lower and no off-target effects. We would have an enhanced target specificity. We would have a smaller protein size, which would make it easier to deliver it into cells. It should have a broad application area, so we should be able to use it in a lot of different cells, an enhanced base pair editing, different PAM motif or nearly PAM-less sequence, so that would allow us to edit almost every part of the genome. And in total, this protein should have an enhanced CRISPR in vivo efficiency, enhanced specificity, and should be easier to deliver into the cells. Beside from this, and this is possibly the most important part for a commercial organization like BRAIN, is the IP situation. Because it would be great to have a legally secure IP situation, which would then generate freedom to operate for BRAIN and its customers and its collaborators. And of course, this would then lead to lower cost overall. So with all the things that we have discussed on the last two slides, and all the R&D capability that we have inside of BRAIN, we came to the conclusion that we need to do something to bypass the current situation and to screen and develop our own proprietary genome editing tool. So we started with metagenomics. So we went out to the fields and to many places, most of them in Germany, and sampled different habitats. For example, we went to a pond, we pulled out a few liters of water, sampled a few liters of water, then isolated all the microorganisms that are inside of this habitat, so inside of the water, isolated their DNA, sequenced their DNA, and then are able to evaluate the genomes of all the organisms that are present in these samples. On average, with our newest sequencing technologies, so using next-generation sequencing, we are able to identify around about 10,000 different species in every sample, and then we are able to evaluate their genomes. So in total, we sampled over 30 different habitats. We generated more than 2,500 gigabase pairs, so billion bases in DNA, Just to put this in perspective, this is just plain text files, so no figures, no pictures inside. And this consumes more than 2 terabytes of storage space on our hard disk drives. We have quite diverse habitats, so habitats with different pH levels, different temperature levels, solid and liquid ones, etc., etc. And in total, in the genomes that we were able to evaluate, we found or were able to identify more than 100 million genes. On top of this, we did functional annotations, so we searched for little amino acid stretches, which are functionally characterized, to search for functions related to genome editing tools like DNA binding domains, DNA targeting domains, and so on. Based on these metagenomic samples, we then build up a pipeline to identify novel CRISPR genome editing tools. The first 2 pictures shown here on this slide are showing the parts of the pipeline that we've already discussed. So the metagenome sampling, the microorganism isolation, the DNA isolation, and the sequencing. And then we set up a proprietary bioinformatics pipeline, so an in silico-based pipeline, where we are able to identify novel CRISPR-Cas genome editing tool using a computer-based approach. Then we used these sequences that we identified in silico and tested them in the lab and selected the best one. And with those, we did protein engineering on top, meaning that we combined several sequences to add up their positive effects. And then on top of this, we did protein engineering on an in silico-based manner, so we changed single amino acids with positive effects. In the end, we were able, by doing this, to generate one sequence with a really high specificity and genome editing activity. And this is our prime candidate at the moment. So this is our BEC protein, so the brain engineered Cas protein. What is brain engineered Cas protein? So the brain engineered Cas protein is a novel class 2 engineered Cas protein developed using a metagenomics approach and a protein engineering approach on top of this. It is a non-Cas9 and non-CPF1 protein, so it is not subjected or not affected by the ongoing patent war. It was designed by metagenomics and engineering, so it has no native origin, so you will not be able to find this protein anywhere in nature, and therefore it should be less immunogenic compared to the classical genome editing tools. Furthermore, we were already able to show that it has a different mode of action compared with Cas9 and CPF1, and I will show you 2 examples of this on the next slides. What are the strengths of our protein? So we have already done experiments and were able to prove the activity in various bacteria, fungi, and yeast cells. On top of this, we have comparable or even better results when we compare the results to Cas9. We were able to show first activity in plant cells together with our partner and are currently evaluating these results to show that we are able to use the BEC protein for genome editing in plants. At BRAIN, we are currently testing the BEC protein in mammalian cells and mammalian cell lines. And also, we are looking at other areas which are not currently in the main focus of BRAIN. For example, based on this new mode of action, our protein could be very well suited for the diagnostics market or diagnostics applications. So what is activity? To show you what activity and genome editing means, I would like to show you 2 examples. For the first one, we used Saccharomyces cerevisiae. Saccharomyces cerevisiae is a yeast strain that is a classical baking yeast, which is also used for brewing like beer and wine, so quite important strain for biotech applications. And what we did here is that we knocked out a gene of the Saccharomyces cerevisiae strain using the Cas9 nuclease and in comparison then to our BEC nuclease. The special thing about the experiments that we've done here is that we knocked out a gene, the gene is called RDA2, which leads to a change of the phenotype of the organism, which means that the wild type gene, so if the gene is still intact, has a white phenotype. And then, if we're able to knock out the gene, the phenotype changes from white to red. So it's really easy to monitor if the genome editing has happened or not. The results are shown on the left side, and what you can see is that in the negative control, first with Cas9, you can see a plate full of white colonies, and then when we activate the Cas9, the colonies change from white to red, which shows that we were able to successfully edit the genome of the Saccharomyces cerevisiae strain. Then we did the same experiments using our BEC protein, and what we surprisingly could see there is that the results look quite different. So negative control, similar results, plate full of white colonies. Then when we activate the BEC protein, we see a huge decrease in the overall cell population. So around about 95% of the cells are just gone, wiped out by the BEC protein. But those cells that are able to survive the treatments, they are edited in a highly efficient manner because over 80% of the cells are showing the knockout phenotype, so their phenotypes change from white to red. And if you compare this to the Cas9 experiments, the efficiency is quite comparable. So in both cases, we have around about 80% of edited cells. So different mode of action, different results, but the important part, which is the editing efficiency, is comparable between Cas9 and our BEC protein. For the second example, I would like to show you a different strain, which is an Aspergillus niger strain. This is a higher fungal system, which is also a strain that is used in a lot of biotech applications, for example, for protein and enzyme production. And what we did here is that we knocked in a gene. And for this, we also used a special gene. The gene is called GFP, which stands for green fluorescent protein. It is originally from a jellyfish. The special thing about this gene is that when you illuminate the protein with UV light, then the protein lights up in green. So also here, very easy to detect. If we have the wild type strain, it doesn't light up. If the GFP protein is inserted into the genome of the Aspergillus strain, then we could see a green fluorescence when we put it on a UV table. Results are shown on the left side, and they are quite similar to what we already saw in Saccharomyces cerevisiae. So we have a negative control where we have a plate full of colonies. Then when we activate the BEC, we have a huge decrease in the overall cell population, and just 3 cells are able to survive the BEC treatment. When we put the plate on a UV desk, you can see that specifically those 3 cells are lighting up. And then if we transfer one of these colonies to a different plate, let it grow for a few days, and then put it on a UV desk again, you can see that all the colonies on this plate are showing this green phenotype. And this shows that we were able to edit the genome of the Aspergillus niger strain. Here we have an even higher editing efficiency compared with the Saccharomyces cerevisiae strains of more than 90%. And this is also better than the results that we were able to achieve with Cas9, where normally we have editing efficiencies between 70 to 80%. So in total, what we could show with these examples is that our BEC protein is a highly efficient genome editing tool that is comparable or even better than Cas9, and it can be used in a variety of different organisms. Where are we using the BEC protein at the moment at BRAIN? First of all, we are already using it in our TMS business, which means that we are using it together with our partners or our collaborators or our customers in several projects. Big advantage of our technology is that we have the big USP of freedom to operate. So we are allowed to use this technology in comparison to all the other CRISPR-Cas genome editing tools on the market. We're also using it in our incubator pipelines, so in our internal projects, for example, in strain development projects, where also, of course, the freedom to operate helps us a lot to use this technology. Then we're working together with our exclusive partner in plant cells, where we already were able to show that the BEC protein is active in plant cells. And currently, we are evaluating the genome editing activity and efficiency in those plant cells. And then we would like to also give sub-licenses together with our partner to other agriculture companies. At BRAIN, we are currently focusing on mammalian cells mainly, which means that we are working on different cell lines, human cell lines, and the experiments are still ongoing to prove the activity and the genome editing specificity of our BEC protein in human cell lines. And we are expecting results by 2022. It takes a little bit of time because human cells are the most complicated ones to work with, we are working in them already. Most of these experiments are done by BRAIN, but some of the minor projects are already outsourced to our partners, where we are working in close collaboration to prove the activity in mammalian cells. Beside from these main focus areas, we are also doing several other studies in parallel. For example, we are elucidating the mode of action, so how our protein is actually working. We are working on an RNP system, so we would like to produce the protein and then work with the protein directly. We are doing in vitro studies, multiplexing studies, and also we are working in different organisms or alternative organisms because we would like to have a broad application space for our genome editing tool to work with it in a lot of different organisms. But also we have more. So even though BEC is our main focus at the moment and it's the prime candidates that we are working on, we have found a lot of more sequences in our metagenomics treasure box because there's not just one sequence inside. There are hundreds of million sequences inside, and we were able, by bioinformatics, to identify already around about 2,000 class 2 CRISPR nucleases. We were not able to screen them properly yet, but we will do so in the future. And we have already selected 15 candidates out of these sequences that could have a huge potential for BRAIN, and have already submitted an IP application to protect the sequences, and are currently developing and testing their activity. Furthermore, we are in speaking terms with additional partners to explore and exploit these metagenomics treasure boxes. And we would like to do this together with a partner to accelerate the detailed screening, so the timing. We would like to share costs with our partners. We would like to gain additional application know-how from our partners. And in the end, of course, we would like to have a faster path to the market. Last but not least, all this research that we are doing and all the monetization that we are planning is backed up by an IP strategy. So what we already have is that we have several single patent applications that we have applied to secure our BEC protein and some sequences around it. We will back this up by additional application IPs in application areas where we have the most interest in. And of course, during all this research and development we've done, we've gathered a lot of knowledge about our BEC protein, and some of this we will keep as a trade secret inside of BRAIN, and this will never go public. What we wanted to achieve there, what we would like to gain there, is that we would like to have a broad application space for the protection of our sequences, and then backed up by the additional application IP. We would like to have a fast international reach and a unique position for BRAIN with the USP of our sequence for us and our partners and collaborators. Last but not least, of course, what could be the commercial impact of this? First of all, big selling point is freedom to operate. So we are the only ones that are allowed to work with this protein. So we could use this in our TMS business, but also in our internal projects and our collaboration projects. And then, of course, we are able to monetize this by the generation of revenue, for example, by tech access fee, license royalties, license revenues, royalty potential, and in the end, also the potential to spin this out as a newco to mainly focus on the development and further application fields for our BRAIN Engineered Cas protein. And with this, I would like to close my part of the presentation. Thank you for your interest in the more scientific part and on our BEC technology. And I would like to hand over to my colleague Lukas, who will give you a more business and commercialization focused insight and what we are planning for the next few years.